Hi. Hi, Adam. How are you doing? Hurt my finger today and it's really bugging me. Good. What have you been doing today? I have been recording one of my long form videos for the course that I will be offering in the Easter. I have just tutored two one-to-one clients back to back and then had my group tuition session for my year 12s. And now I am jumping on here to help any of you guys who might need some help. So that's what my day's been like. Obviously, and I picked up the girls in between that and cooked some dinner as well. So been a pretty busy day today actually. Wednesdays are pretty busy. But yeah, it's been a good day. How about you? How's your day been? Anything exciting happening? No, I haven't done any new art things. Oh, actually, that's a lie. I did do one yesterday. Hi, Carol. Um, I did a little drawing yesterday because I was looking at it was like a new way to draw a person's standing position, but it was just a quick one, and then the girls wanted to color it in, so it's not that great. But I've not had time to do that because I've been doing. I did the muscles workbook, and now I'm building the hi. I'm building up the course now. That's where my focus is on my group tuitions, my one-to-ones, and my course at the moment, trying to get all the stuff that I need to do. And obviously, there's still more study guides to get done. But yeah, it just really depends. If I have the time, then I do try and do it. But I was away at the weekend. I'm away again this weekend, which makes it a little bit less easy. Also, the girls are busy playing other things. Quite like drawing with them. My eldest is really good at art. And my youngest is getting much better as well. So they'll be the ones doing the drawings for me. Yeah. Welcome, guys. If you are joining me, please do ask any questions if you have any questions. They are very artistic. You're absolutely right, Carol. They are. Bless them. They've got art tomorrow, so hopefully they'll have fun. Last week, what did they do in their art class last week? Last week they made little penguin ice skating sculptures. It was really cute. And they had little magnets that allowed them to move it. So if they moved one bit underneath, it then allowed for the other one. But I didn't teach the science behind that and the magnets. Not yet. What were we doing the other day there? We were walking somewhere and I started teaching biology and I was thinking, oh, can I not just have a conversation that's not about biology? My 6-year-old was like, come on, mummy, what? I can't think what it was we were talking about. It'll come to me if I stop thinking about it. We do that quite a lot. Like, stop thinking about whatever it is you're thinking about and it'll come to you what you want to say. So yeah. But it's a very interesting skill to learn. You're unfortunately doing EdXL because I remember asking you about transcription. That's okay. You can still ask questions. EdXL is very similar. Are you doing EdXL B or EdXL A? I don't actually know what the difference is with EdXL A in comparison. I choose EdXL A. Interestingly, you said that EdXL A-level. Let me have a look. Biology. EdXL A. Let me have a quick glance and see what it says. What's the difference? Oh, you're doing the Soldiers in a Field one. Oh, I should know it's that one. It's a funny one. Different skills to different ones. Obviously there's different examples out there. I don't know what it is about that one, but I feel like I don't like it. We've just started a new topic on biodiversity and plants. The key thing is a lot of the biology content overlaps. So a lot of it is really good at overlapping, but it's just making sure that it's specific to what it is you need to know. Let me look at the biodiversity. So things like, I think your biodiversity Simpson's index equation is different. So topic 4, biodiversity and natural resources. I'm guessing it's that one. Yeah, so your Simpson's indexed equation is slightly different to the one that OCR uses. Little things like that you've just got to be careful of. But going through, so variety of life, terms biodiversity. And again, see, you've got to know about endism, whereas they don't need to know that in OCR. Niches, they go into more detail on niches here. Natural selection, that's pretty much the same. Hardy-Weinberg, same. Formation new species, so speciation, same. Classification, process the critical evaluation of new data, pretty much the same. Recognize organelles, same. Polysaccharides, beta, cellulose microfibrils, same. Cellulose microfibrils, same. So yeah, it's pretty much the same. Is A-level biology really hard, or is it not that? Okay, so A-level biology is difficult, it's challenging. However, if you keep on top of the content and you take the time to do the revision and the practice, then it's manageable, but there's a lot to learn. So depending on which exam board you're doing, that is going to make a difference as to whether the questions are slightly easier or more accessible, or whether they're harder. But every exam board is a level 3 qualification, so they're all the same level of a qualification. So it doesn't matter whether you do an AQA one, you do an OCR one. What's happening is it's about what that exam board has focused on, basically, and they've said, right, this is what I want to look at, these are what we're going to push. So there's things in OCR that you have to know which you don't need to know for the other ones. And like I was just looking for else there in the Edexcel A, they've got stuff that they have to know, the extra bits for their course, which don't need to know for OCR. So it is challenging, it is hard, but it is accessible if you work at it, but you just have to really work at it. Biology is one of those subjects that you can't just read it and learn it once and get it. You have to do it again and again. You really have to fully understand it. You can't pass the exam at a high level without having the knowledge, and that's why it is a subject that you will have to get those foundational skills sorted before ideally moving on to year 13. So a lot of students, if they're starting year 13 without having a strong foundation, it's going to be really tough. Just wait until, yeah, exactly. You're absolutely right, else you can't just start waiting until year 13 start revising. It's the worst thing you could do for biology, especially. And students still think that they can cram and get the grades that they want to get at the end, and they just can. And I find it really fascinating. So I jump on here and I do these sessions twice weekly now again. Ideally it's only going to be a Wednesday, but I do seem to jump on a Monday if I've got time. But the one thing I've noticed is, because I do the scrolling myself, and the reason I do these sessions is, one, to maybe stop you scrolling for a little bit of time and give you some knowledge, but two, because that addictive scrolling that you get through using these actually stops you from engaging. And I find it fascinating when I'm scrolling and I'm looking at these people, when I see students who are like, come and revise with me, and I get the come and revise with me as a body doubling process. However, I know that that wouldn't work for me because I get too distracted. I'd be looking, I've done it in the past where I'm like, oh, I'll do a bit of work and I'll just pop on live as well. And then I get so distracted, I just end up chatting and I don't actually do the work. And I wonder for how many of you, the fact that your phone is there, the fact that social media is so accessible, how much of you are actually able to really fully focus on what it is that you're studying and process the information that you're getting. Even I did biology A level and got E's most year and revised for 2 months straight and got a B. So I guess you can. Well, see, this is an interesting one. I'm not saying you can't cram. That's not what I'm saying. But what I am saying is you were able to get grades for most of the year. So you were actually hitting E grades. And so the fact that you managed to get up to a B is fantastic. You did 2 months straight of studying. Just think what would have happened if you'd actually been doing it all the way along. So what you have to realize is there's so much to learn biology. You have to keep on top of it as you're going. I'm not saying you can. I'm saying it's not a good idea to do that because it is challenging. And for those of you who are stuck on the likes of a C grade, what I would say is first you need to identify what it is that's holding you back. So are you not giving detailed answers enough? Do you not know the content enough? You'd be surprised how many students say to me that they are really confident and know and happy with the content that we've been learning. And then I'll ask them a question and they don't have a clue. And it's not that they don't know the content, it's because I ask it in a way that's not quite the same way that they've expected or seen in the exam questions that they've been exposed to. And because of that, I'm identifying what they don't know, so I'm filling those gaps. So it's about finding out what's holding you back, working on those skills to be able to improve to the point in where you get it in the exam. Do you think going from combined a GCSE to A level is a huge jump? And do I need to learn? No, you don't. So it doesn't matter whether you're doing combined or whether you're doing triple science, just so you know, as long as you are hitting ideally level 6s. So most sixth form colleges won't accept you onto level 3 qualification, especially for STEM subjects, unless you're getting a 6, depending on the college. So for example, Hales Road in Cambridge, say they'll take you at 6, but really they want a 7 or higher. And so it's just about getting that foundational skills. Combined science gets you to where you need to be, and then your teachers at A level should be taking you through that extension that you need. So no, you don't need to do triple to be able to do A level, but just make sure you've got strong GCSE knowledge moving forward. Your weakness is exam questions and the applied questions. So that would be doing lots and lots of practice, and then figuring out, again, when you do the practice, don't just memorize the mark scheme. So many students will do, say they've done lots and lots of practice where they're looking at the questions, they are practicing the questions, they're answering the questions, but what they're then doing is they're marking it based on the mark scheme, and when they get it wrong, they are memorizing the mark scheme answer. That's not the right way to do it. What you need to do is you need to convert it into a understanding for yourself so that you have the knowledge to then be able to put that across on paper if they ask you a similar or slightly different question but around that same topic area. Els did combine to A level and they kind of touched on a triple thing such as RNA. Yeah, there's not much of a gap there. Have you gone over the A level biology papers in 2025? Yes, I have. I haven't done them online. I won't go do them online because they shouldn't be available to anyone apart from those students who are in schools doing their mocks. So it's not one that I will do because that's not fair on the people doing them as mocks. Oh, if you're predicted an AA at the moment, so you're absolutely fine. I wouldn't worry at all. And A-level biology, if you enjoy biology, will be a great subject to study. Obviously, I'm a bit biased, but I do love A-level biology. It's why I do what I do. But since got combined, feel like the other people will put me down. Really good at bio, but since doing combined, feel like the other papers will pull you down. So what, you think your chemistry and your physics papers will pull you down? I have combined higher science mocks for GCSE in 2 weeks. I try to revise but can't learn. What should you do? What I suggest, if you are struggling with your content for GCSE, when you say you try to revise but you can't learn, how are you revising? This is another thing, that as a student, not knowing how to revise or what revision works for you really is one of those skills that if you can nail quite early on and do the right stuff at the right time, and it's not like doing hours and hours and hours, that's a waste of time. It's about doing the right kind of revision that works for you in the amount of time that you've got. You got a B last time, but you were 3 marks from an A. Do you think the real thing I can get an A? Absolutely, 100%. If you were sitting at a B grade, as long as you are evaluating what it is you did that stopped you, 3 marks away from an A is a really good place to be, but you need to make sure that you're doing the revision and the content and getting that understanding that you can then put that across on paper. So absolutely, it's possible in the real thing. If you're sitting at a B right now, 100% is possible. The kidneys still haunt me. Oh, worst topic ever. Or the immune system questions. Do you know what? It's a funny one. I had students who absolutely hated the immune system, and no matter how many times I'd go over it, they really struggled with it. There's always going to be some topics that you hate and some topics that you love, and it's about finding how to love the ones that you hate, which is the hardest topic area, the hardest thing, I would say. How should you revise? I try to look through the content and do past papers. So I would say at this point, because your GCSE is, I would say do the question papers, try and answer the question papers, and then try and mark them, but mark them really harshly. Figure out what you don't know. Why didn't you know it? Even when you read the mark scheme, do you actually understand what the mark scheme is saying and what it's saying? And then use that to guide you on your revision. One thing I don't get in bio is the practical with a lamp pondweed and measuring something. Adam, hold on, Adam, are you the GCSE or are you A-level? Because if you're talking about the practical with a lamp pondweed, then what you're talking about is the fact that you're shining light for the light dependent reaction, and with the light dependent reaction you're going to get photolysis of water, and that photolysis of water will release the oxygen, and you're then measuring the oxygen. And if you're GCSE, you'll just say you're measuring the oxygen by the bubbles that are counting the bubbles, but that's not ideal. It's not going to give you a good set of results. You'd want to measure the volume of the oxygen. So if you're trying to look at improving your skills, they're the sort of things you want to say, and that's GCSE and A-level. Hi Christina, I have a test in 2 weeks on enzymes, membranes, nucleotides and nucleic acid, biological molecules and cell structure. Any tips on questions you think students lose most marks on? Oh, that's quite a lot of topic areas. Right, hold on, let me think this through. So where do students lose marks? The first thing that they lose the marks on those topic areas is that they don't understand when the question's asking about enzymes, but it's not saying it's asking about enzymes. That's quite a common problem, that they're jumping with enzyme questions to temperature denatures the enzymes, but they don't talk about the bits before, in the fact that if you've got an increase in temperature, that's going to have an effect on the kinetic energy of the molecules. And if you've got an increase in temperature, you're going to have more collisions. Are those collisions successful? So it's about making sure that you are describing what's happening and not just jumping to an end point. So from the first part of the enzyme, what happens at low temperatures would be one of the things. Biological molecules, a lot of students struggle with the calibration curves and the colorimetry for some reason. So I'd say that would be one to focus on. Nucleotides and nucleic acids is huge. I've just actually done today it's an hour and 14 video. I've not put it on. It's not on my channel. It's not available. But I've just done one. I've done the recording today for the course that I'm running on that. Nucleotides and nucleic acid is the one of the fundamental parts of the content that I would say, if you can get your head around that topic area, if you can get your head around how the DNA, the nucleotides, the process of transcription, translation, how that all interlinks to the expression of the inherited alleles that you've got, that's going to make a big difference for any question that they give you. And I know I've given you a bit of year 13 content there, but I would definitely say that they're ones that I would be looking at. And the other thing about membranes is a lot of students get confused with whether you've got a carrier protein, a channel protein, and what type of movement is going on. So is it active transport? Is it facilitated diffusion? Is it diffusion? So when you are describing these processes, like if it's active transport, are you actually talking about the fact that whatever is being moved is going from a low concentration to a high concentration, so against the concentration gradient? So it's that whole thing of students assume that they have told the answer to the examiner, to the teacher, but they've implied it. So a lot of practice should be going down on how can you actually be explicit in your answers, giving enough detail to get the marks, rather than just you implying it and making me work for it. So my students hate it because they're like, Christine, I said that. And I'm like, no, you didn't. I was like, you've implied it, but I don't know if you're talking about that or if you're talking about something outside. Like, you have to be explicit in your answer. Right now, what you're doing is just doing content from my videos. Thank you for watching my videos. I appreciate that. And your textbook. Then I just do past paper questions. What I will say is that my marking might be off, and on questions I'll write something totally unreliable or irrelevant. I'm kind of guessing what you were trying to say there. So what I would say, if that is the case, so when you are marking, what you should be doing is in the mark scheme, when you use the mark scheme, highlight the key words from the marking point and then use that to guide your answer for how you can make it more specific. Now, if you are giving irrelevant information, and that happens a lot with students, that will actually cause you harm. So that's the thing of people who cram, because somebody said earlier, oh, I did lots of cramming and I managed to jump up grades. Absolutely amazing if you manage to go up those grades. However, you'll never get up to the A star random bits of information. So you need to take time on reading the question, dissecting the question to understand what it is that is actually asking you. Students came out of the exam, whichever year it was, and they came out and they were like, oh, Christine, there was nothing on this, this, and this. And then I looked at the paper and I was like, yeah, there was that, and that. And they're like, that wasn't about that. And I was like, yeah, it was. They're like, how did you see that? I was like, well, because I read the question. Obviously, hindsight is a wonderful thing, and I have time to read the question without having the pressure of the exam situation. But a lot of the time, students don't actually understand what the question's asking, and so they therefore start talking about a whole lot of random stuff that's not relevant, and that's where they lose marks. You also do pretty bad at unlike module 4, except for the communicatives, the other two topics that you kind of hate. And that would be where I'd say keep going over those topic areas to make sure that you're trying to find the bits that you hate. Why is it you hate it? What is it that you're not understanding? Ask the questions and then figure it out, so that if they ask a question about that topic area, you're not thrown off. And just be aware there'll always be questions in the exam papers that are difficult and you're not able to answer, or you're not sure. As teachers, we're the same. We kind of look at the papers and go, what were they actually wanting in that question? Sometimes it's not clear, and so that we have to work for. I know why I might not know this, but do you know any questions or patterns that always come up in GCSE tests? Yes, enzymes always come up. Respiration always comes up. It's a funny one when you're looking at the papers, that the topic area, everybody can do predictions. Everybody can read the information that's out there, have a look through, and they can do their best guess of what they think is going to come through. The GCSE papers are pretty standard. If you have experienced as many past paper questions as you possibly can, i.e. all of them that have been available since it first came out, then you will have experienced all the regular things that come through. Looking for only the patterns is going to either be an amazing day for you, where you've predicted right and you've only studied the bits that are right, or it's going to be the worst day possible because you haven't learned all the content. So I would say when it comes to GCSE, you've just got to learn it all. You can learn the fundamental parts. So have a mind map, for example, for the topic areas, and have just the key words and have them in your head and then apply them to the question. But just learning the things you think will come up each time will never get you anywhere. Be honest, what method for revision would you realistically recommend for biology to really improve the grades? Is it past paper questions or more so content, or a mix of? It's got to be a mix of both. You can only do so much by doing the past paper questions. So that exposes you to the types of questions, the style of questions, the way in which they will ask you the content, and it will give you an idea of what they accepted. And this is the important part. The mark schemes that are there are not for you to memorize, because the mark schemes were valid the year that that exam was sat, because those examiners got together and agreed on that official mark scheme. So when the exam is being written, the exam questions get set, the mark scheme gets set at that point for the assumed answers that will be given on in the exam for those questions. And there will always be some bits where there are extra points that you will get. Once you have sat the exam, once there has been a set of them that have been marked, what will then happen is a team of senior examiners will get together, having marked these papers, and they will then go through and say whether they'll accept something, whether they won't accept something. And that will then become the allow, do not allow, ignore, extra bits in a mark scheme that you see. That is valid that year that it was sat at that point. It's not valid for every other year. It's not a, there is your cheat sheet and that is what you need to memorize. That will not work. You need to know the content. You need to know what is actually going to help you moving forward. So you have to do both. You have to learn the content, and you have to also do lots of practice paper questions, as many questions as you possibly can, without you just focusing on one, because obviously you've got 3, if not more, A levels depending on how many that you're doing. So it is a try and do lots of different things. Try and focus on what you don't know. Use the past papers to identify what you don't know and go back and revise that. Oh, amazing. You've gone from an E to an A. That's awesome. That's so impressive. Well done. How should you revise biology? It really just depends. Everybody is so different. So it's got to be doing what is right for you. And so for me, I know that I have to do a lot of repetition to remember things, to get it ingrained in my brain, the content, so that I can regurgitate it. Now, I know OCR inside out, but I still, even though I know it inside and out, even though I've got my YouTube channel, even though I've got my videos that are there, I still go back and double check the specification. I still make sure that I am relevant and I've not confused anything in my head. Because I tutor lots of different exam boards, I also need to be aware of that. You are studying other subjects too. And sometimes, for example, some of you will choose psychology and biology because they work really nicely together. But if you start answering your knowledge from psychology in your biology exam, you're going to lose the marking points. So you need to make sure that your head is, I'm in biology, this is what I need to give. It's the same when students are doing chemistry and biology together. When they start giving me the chemistry answers, they're not going to get all the marks. And so therefore, you have to be careful that your answer is biological and relevant to what it is you're being asked. GCSE is so easy because it's memorizing mark schemes. I would agree that technically it's more memorization than it is application, unless you're going for those 8s to 9 grades. I wish you could learn A level biology like you can at GCSE. I think you can. It's just that there's so much more content and so much more depth to go into that it makes it more difficult. Why is AQA A level biology so hard? All the A level biologies are hard in their own right. They just have different ways of being difficult. So my main exam board, so you said, is my main exam board OCR? So what happened, the reason I made OCR resources is when I first left my job where I was director of student progress. So I'd worked my way up from being classroom teacher to being head of biology to being deputy head of lower school, head of house, director of student progress. So I'd worked my way all the way up to a senior level within the school. And then I had my 2 children. So I have 2 young daughters. And when my daughters came along, my life had to change and had to be supported around them. And so I ended up leaving the job that I was doing, that I had done for the last 11 years as within the last school that I was in. And I started tutoring, and I was tutoring my niece at the time. And so I was like, well, I'm tutoring my niece, I'm quite enjoying it. Maybe I'll start doing this as a role. And I went, and I've been teaching the IB for many years. And then I went to support a private school in Cambridge, and the exam board that they were teaching was OCRA. And so I hadn't taught OCRA for such a long time. I mean, the last time I taught OCRA was when I first got into teaching in 2007 till 2010. I left 2011, I left my first school. And so when I was first teaching, I'd done OCR, and then I hadn't done it for such a long time. I'd done the IB. And so when it, and A level, because we did A level, AQA A level with the IB, and then just full IB. And then I taught the IB sports science as well. So I hadn't taught OCR for a while. And when I went to the private school and I started looking and was teaching them, and I can teach any of the exam boards as long as I have the past papers and the specification. And when I started looking for, right, where can I direct my students to find resources, I couldn't find any OCRA resources out there. And so I started to build them. And as I started to build them, I started on TikTok. What happened was, when I was on TikTok, people were coming. When I finally managed to get on the live side of things, people would come to my lives and be like, oh, have you got a YouTube channel? I was like, no, I don't have a YouTube channel. So then they were like, oh, you should go live and do a YouTube channel. So then I started the YouTube channel. So I took 6 months out, no pay, to focus on building the YouTube channel for OCRA. So is it my main exam board? Yes, because that's the one I decided to build the resources for. I also teach AQA privately, as well as the IB. So it's just that the channel was built around the OCR resources. And so I have consumed myself with OCR. I know that one inside out. I can pick which specification module you're looking at, etc. But also I can do it to an extent with AQA. I have to remember, oh yeah, well that one is there. For example, recombinant DNA is the same as manipulating genomes for OCR. I forgot to also ask your advice on common slip-ups with questions on mitosis and meiosis. Now that's an interesting one. So mitosis and meiosis is, they will throw a novel idea at you on that one with regards to, do you know whether it's a haploid or whether it's diploid cells that are being produced? So if you're doing mitosis, you should know that you are getting genetically identical daughter cells. And because you're getting genetically identical daughter cells, if you start with a diploid cell and you end with a diploid cell, that is mitosis. This is where students fall and slip up quite often. If you start with a haploid cell and you end with a haploid cell and some form of division has occurred, that is still mitosis, because you are ending up with genetically identical daughter cells from the original cell. So students seem to think that if you've got a haploid cell, that haploid cell has to be produced through meiosis. That is only true if you started with a diploid cell, 2N, which then was reduced down to a haploid cell, because you're then reducing the number of chromosomes in the cell. So that would be the major slip up, I would say, for mitosis and meiosis. The other thing would be, if you're looking at meiosis, then knowing that there is obviously 2 reduction divisions, health and disease. What part of the health and disease can you not wrap your head around? What bit specifically are you struggling with? Thank you, by the way, for all the likes. Oh my word, 1.6 thousand. Thank you. I really appreciate that, guys. Thank you. Yeah, what part of the health and disease? I don't know if there's like a delay on the YouTube chat. If there is, then I will answer when I see the questions come up. But is it that you're not getting the different types of cells? So like the B cells to the T cells? Is it that you're not sure on which of the diseases and how that would affect it? What is it that you're actually struggling with the most? That's the bit I'd say you focus on. And that's what I mean when it comes to revising as well. If you're revising, actually knowing what it is you need to revise. So saying I'm struggling with communicable diseases is such a massive topic area. So what is it that you're actually struggling with? Which part is it that you do not get? Do you actually know which bit that you don't get? So the action of a pathogen. Okay, so when you're talking about, are you AQA or OCR? Because when you're talking about the action of the pathogen, what they're looking for you to note is which pathogens there are and what diseases they cause. But I feel like the way you've worded that was the action of the pathogen, that you are AQA. But let me just confirm that. I've got the specification open. I always do. I have both the specifications because I tutor an AQA student, and before I do my group tuition sessions. Your OCR. Oh, hold on. When you say the action, see, now I'm going to get the OCR one open because now you're making me question my knowledge. So if you're looking at the action, is your wording that you said the action of the pathogens? Oh, that makes more sense. Okay, the action of the phagocytes. I was thinking that. I was like, that doesn't make sense to me. Why? And especially if it's OCR, I was like, that wording's not in the specification in that way. Okay, that one I can help with, and that one I'll give you. Give me a second. I've got a really nice, she says, little animation that I do on that one. So let me just get it up and I'll bring it across. I was starting to worry there. I was like, oh, I need to go back and double check. I've maybe got this wrong. And this is the thing. I do go and double check. I never believe that I remember it all, because actually that's not the right way to do it. Even though I have been teaching this for a number of years, and even though I do know what I'm talking about, it's always good, and I should be going and double checking because that's important. So if we talk about the actions of a phagocyte then, so it's important to know a phagocyte is a cell that's going to do phagocytosis. So if this is my pathogen here, so here I have a bacterial cell, and I can tell that because if I zoom in, we're going to zoom right in. I love this. The fact that I can zoom in and actually still see the detail in, ooh, crumbs, went too fast there, in my diagrams, it makes me really excited, really sad. And so we know that a phagocyte is going to engulf and digest the pathogen down. So what is it that we need to understand? Well, we need to understand it's a cell. It's a cell that has a cell membrane, a cell that has cytoplasm, a cell that has the cytoskeleton, a cell that has lysosomes that contain lysozymes, a cell that has a nucleus that contains the genetic information. So this is a neutrophil, and a neutrophil has got a lobed nuclei or nucleus, and so therefore we can distinguish that from other phagocytes based on this lobed nucleus. So what is actually happening when we do phagocytosis is, do you understand that the phagocyte is attracted towards the pathogen by the chemicals that the pathogen is producing? So therefore the phagocyte is going to recognize the pathogen as non-self cells. So the fact that the phagocyte has got the cytoskeleton that allows it to move means that it can move towards the pathogen. So it's detected chemicals, and for those of you doing AQA, that is a form of chemotaxis, where the cells can move towards the chemicals because of the attraction to the chemicals that are being produced. So the phagocyte is going to engulf the pathogen. Now, you can see here that what's happening is it's kind of sending out these extra extensions. This engulfing process is where the phagocyte will send out these arms. They're called pseudopodias. I love that word. And what's going to happen is these arms are going to come out and they're going to surround, engulf, and basically pull in the pathogen into what's known as a phagosome. So the fact that this is an example of endocytosis, we're taking in this pathogen, we're going to put it into a little compartment, and that little compartment is a phagosome. When do we learn about chemotaxis in A-level biology? For AQA, I'm in year 12 and I've never heard of that. So that's the responses. It'll be a year 13 content for you. So what we're going to get is, you can see that these two parts of the cell are coming together, and as they come together, because they're a phospholipid bilayer, because they're made of the same stuff, what can happen is they can fuse together, and when they fuse together, what that then means is you have this pinching away. So we have the plasma surface membrane, which is intact, and we now have this pathogen contained within its own little compartment. You're very welcome. So then what we need to understand is, what we can see here is these blue dots inside the cell are my lysosomes. Now my lysosomes contain lysozymes. Again, a lysosome is a vesicle, a membrane surrounding these enzymes that can do the digesting. So we want to make sure that these enzymes are contained inside these lysosomes, and then what happens is they will be able to move because of the cytoskeleton. They can move and they can then fuse with the phagosome. And when the lysosome and the phagosome fuse together, you are going to get a phagolysosome. Now, one of the biggest problems most students have in the exam is that biology is full of so many big words. We've got a phagosome, we've got a lysosome, we've got a phagolysosome, we've got gluconeogenesis, we've got glycogenolysis, we've got glucagon. We've got all these words that sound so similar, and if you get them wrong because they're key scientific terminology, you will lose your marks. So one of the other things I'd say is, as you're doing your revision, is try to make sure you are specifying those key words every time you give an answer. And if you're getting it wrong time and time again, have a way to identify that that word is not stuck in your head. So you'll hear me say things and then I'll listen to myself, and if I've got it wrong, there might be a slight processing delay, but if I get it wrong, I do listen, I hear it, and I try to correct myself. So it is really important that you do that yourself. Now, whether that's in front of a mirror, whether that's to a study buddy, whether that's recording yourself and then listening to it back, these are all forms of revision. So if you are someone who wants to find a way to revise and maybe doesn't want to post a video, then don't post it. But you can record yourself and then listen and watch it back and say to yourself, oh, did I actually get that right? Really listen to what you're saying. It's one of those things that's really interesting. I watched a video on it from a person who is a professional speaker. They help you in improving your communication skills. And they say that you should actually, if you're trying to improve your communication skills, what you should do is you should record the video like I do. And then you should listen to the video back without being able to see what you do. So I know for me, I stumble sometimes and I'll say okay, or I'll say like the same words over and over again, and they are like my filler words, like they are like my filler words. They're my filler words. They're my words that's my brain's trying to think of those things that I want to say, but they're not coming because I'm going so fast, because I'm like as quickly as I can. I'm like, get this information out. That is what I do, and that's something I know that I do. And so when you listen to yourself, so if you record yourself but you don't watch yourself, you listen to yourself, you will hear the words that you are using, whether they're correct words or whether they're the wrong words. Now, get over the cringe. And what I mean by that is, if you make a mistake, I will make a mistake when I say things sometimes, and that's okay. It's okay, because I know what I meant, and I know I will write the right thing down. And I've had to train myself because I'm from Scotland. So for me, a lot of the words that I use aren't words that potentially I would have used in the past. So when I first came to England to go to Cambridge to study and do my PGCE, I was teaching about the Bunsen burner. But I didn't call it the Bunsen burner. I called it the Bunsen burner because that's what it was to me. And I had to say it over and over and over again to train myself out of using the wrong terminology. I would write it the right way, but I just would say the Bunsen burner rather than the Bunsen burner. And it was interesting because today I'm walking home with my girls and their friends. They said a word and I couldn't work out, oh, what was it? It was putty. We were talking about what to get for a birthday present, and they said putty. Oh, this person really likes putty. And I was like, oh great, they like putty. And they're like, that's not what we said, it's putty. And I was like, well, that's what I said, putty. And they're like, no, putty. And it's whether you say graph or graph, like whatever you say. And I can't even do that without putting on a posh accent. Like, it doesn't matter how you sound. Nobody is assessing you on how you sound. They're assessing you on your knowledge. They're assessing you on your ability to show your understanding and to apply your knowledge to the exam question in front of you. So do make sure you are practicing using the correct terminology, which is where this all started with, when you are answering the questions, and make sure you're writing that down in full so that you are making sure that you've got the right terminology for the exam question. So if anyone is sitting at a B and not getting up to the A star and they're wondering what it is that's holding them back, that is what you need to do. You need to get your keywords down and make sure they're the correct keywords for the question at hand. You are very welcome. Can you also go over the B and T lymphocytes and the specific immune response? Yes, I can, but I haven't finished yet, even though I went on a little bit of a detour. The lysozymes digest and destroy. The release of the cytokines are your chemical signals. These are molecules that are going to attract more phagocytes to the area. So if you're going to attract more phagocytes to the area, you're therefore going to engulf and digest more of the pathogens that are there. Because remember, pathogens like bacterial cells can replicate at a very fast rate. So they do binary fission and they can go from 1 to 2 every 20 minutes, 30 minutes, depending on the actual pathogen that you're looking at. So it's important that you know these extra bits of information. Right. if I was going to do the B and T cells, let me just, hold on, pull in a quick slide on that one. She says, whether or not I've still got it. Oh, thank you. Thank you for the likes. I do appreciate it. This one. This is probably, I would say, the one that I like the most, of just doing it in a quick way. Oh, thank you. I do appreciate that. Right, okay. So let's look at the different ones between the B cells and the T cells then. We just learned this last week. Such a good recap for me. Oh, there we go. Lovely. Right, what's the difference between B cells and T cells? So you should know that your blood cells come from stem cells. Stem cells, your hematopoietic stem cells, are stem cells that have the ability to go through mitotic division and differentiate into whether it's erythrocyte, whether it's a lymphocyte, whether it's a leukocyte, depending on the cells that are needed. So that's the first thing for you to note. You have these multipotent cells. Now, if we have a B lymphocyte, it's a B lymphocyte because it is produced in the bone marrow, but it also matures in the bone marrow. So these B cells mature in the bone marrow, whereas your T cells mature in the thymus. So they migrate from the bone marrow to the thymus, where they then mature. T cells are for cellular response, but B cell, yes, that's correct. So when you're looking at your T cells, you should know the different types. So you've got T helper cells, and the T helper cells are going to produce interleukins or cytokines. You can use either of those as the answer. So they are going to produce these chemicals which are going to signal the activation, for example, of a B cell. Now, the B cell can divide by mitosis and produce different cells. So the two cells for the B cell is a plasma cell, and the plasma cell is the only one that is able to produce the antibodies. So the bits that students miss whenever they're revising this is that you have to remember that to go from one cell to more is through the process of mitosis. So the cell has to be able to divide by mitosis, and then it needs to differentiate. So it differentiates into the plasma cell. The plasma cells are then going to do protein synthesis, and that protein synthesis is going to be done via the rough endoplasmic reticulum, the Golgi, because we are going to produce these antibodies, and these antibodies are going to go into the bloodstream. So therefore, this is going to be released via exocytosis. So it's really important that you don't just think, oh, I'm doing the immune response, so I'm only ever going to be asked about the type of cells. That's not how the exams work. The exams are expecting you to be able to put those linked together. Now, if you are OCR A and you are doing this exam board, they will be short questions, but will expect you to make those links. If you're AQA, the expectation is that you can make those links, and that could come up in your 25 marker on paper 3, or in the extended reading section B of paper 2. So it's really important that you don't just think of the cells or the topic areas as being, I'm doing the immune response and that's it. It's not. None of it's that. It's all about the foundational skills of biology coming through it all. What are we talking about? We're talking about a cell that has DNA within the nucleus. The DNA within the nucleus, that determines whether or not it is going to produce the antibodies. So therefore, these signals these interleukins are going to bind to receptors on the plasma membrane. So we could talk about the plasma membrane. So I can go in any which way you need to go on this. It's important that you can do that too, that you can see those links that come together, because that is biology. It's those links that make it make sense. So the antibodies are produced in, obviously, the plasma cells and released into the bloodstream, and they will then travel around in the bloodstream and bind to the pathogen if they encounter it. Now, when we look at the T cells, you've got your T helper cell that produce the interleukins. That will obviously activate our B cells. We also have the fact that, let me go to that one second. The interleukins can also actually be released to cause the phagocytes to come to the area where they have encountered a pathogen. Now, the other thing for you to note, if you are AQA, you do not call it a T killer cell. These are cytotoxic cells to you. So, and that's where it's important that you know for your specification what is it that's required. So OCR, we call them T killer cells. The AQA, it's cytotoxic cells, and it is important that you double check your specification, that you're looking at these keywords, because if it's in the specification, if it's been listed there, that is the word that they want you to use. And I believe, let me just double check this before I say the wrong thing. When it comes to the B cells, being activated by your T helper cells, cytotoxic cells, is it interleukins? Is what I want to double check for you guys. They haven't specified in your specification, so I think you can use both of them on that one. Now, your cytotoxic or T killer cells are going to produce perforin, or it could be hydrogen peroxide, depending on the question that they want to do. That is going to be used to actually break open the cell. So these chemicals are going to be injected into the cell to kill the cell, which is why they're called the killer cells or cytotoxic cells. They are toxic to the cells. And then the other one that I'll finish on is the T memory cells. And obviously having these memory cells means that you will have a faster response if you encounter the pathogen for a second time. So I hope you have found these sessions helpful. And I hope you join me again next week. I'll be back on Wednesday, as I do. Mondays depends if I'm tutoring or not. So if I'm not tutoring, I will be potentially there on Monday, although I'm building my new course, so maybe I need to focus on that. But I'll definitely be here on Wednesday. So thank you for joining me, and make sure you do have a rest, because I have been talking for a long time. I am going to have a rest now and go to bed. So guys, have a good one, and I will see you next week.